Welcome everybody to our monthly Grow Our Own Timber seminars, or GOOT for short. These are seminars that we have to build discovery related sciences. And today we're very excited to have Miquel, who's going to be talking to us about demystifying AI for drug discovery. So Miquel is CSO and co-founder of Ersilia, and he'll chat to us a little bit more about his background, if that's his plan, and then take us through his presentation. So thank you very much again, Miquel, and over to you.
Thanks, Alasia, and thank you everyone, and thanks for the invitation. I'm very excited to speak to this audience today. So just a little bit about myself. I'm an organic chemist by training, but did my PhD in computational biology, so I guess I consider myself a computational pharmacologist. And today I've been asked to give a sort of an overview or an introduction to artificial intelligence for drug discovery, which is something that everyone is talking about these days, and it might sound overwhelming. So I'll try to do my best really to break it down and simplify it for you. Okay, please, if you have any question while I'm speaking, just ask your questions through the chat, raise your hand, and I will stop and I will try to answer. Also, if you feel I'm going too slow, just let me know, feel free, I won't take it bad, and we can accelerate and complicate things a little bit further, okay? So you see my pointer? I hope you do. So today I'm speaking for Ersilia, on behalf of Ersilia. Ersilia is a young enterprise, a not-for-profit enterprise that we have started together with two molecular biologists, Dr. Eduardo Gaude and Dr. Gemma Turon. Eduardo did his PhD in Cambridge University, Gemma in the University of Barcelona, and we are basically good friends, and we are starting this initiative to bring AI into the laboratories around the world. So today I'm going to speak a little bit about our initiative, and also I'm going to speak about my background work that I have been doing in Barcelona, at IRB Barcelona, in the group of Professor Patrick Alloy during the last couple of years, okay, which is very related to what we do now. So what's the motivation, and why did we start this not-for-profit enterprise with Eduardo and Gemma? Well, I would say we have two missions or two goals, or we are concerned about two facts that are evident these days in the world. One of them, you know it better than I. There is a Western bias. There's a bias towards Western research in biomedicine. These numbers are, I mean, we might discuss about these numbers, but they are pretty clear. They send a clear message. There's a small proportion of drugs in development these days that are being developed for infectious diseases, even if infectious diseases, as you know, represent a majority of the disease burden in low and middle income countries. For example, in the African continent. This situation, we believe, it's not likely to improve if we don't do something about it, because the scientific productivity in low and middle income countries is also these days very low, which is a pity because, as we know, most of the people in the world live in low and middle income countries, so there is huge untapped scientific research that is not really being exploited. So we are a small team. We want to contribute to sort of bring more balance to the situation, and the best way we can think of is by adopting AI methodologies. The reason why we think AI can help reduce this imbalance is because AI is really low cost, as opposed to other technologies in current biomedicine, experimental technologies. And today, most of the AI that is being developed in the world is based on public data. So in principle, there is no reason why AI research, cutting-edge AI research, could not be performed in low and middle income countries. However, and to my surprise, the adoption of AI methodologies, not only in low and middle income countries, but actually around the world, is being very slow. And there are many reasons for that, but one of them is the fact that only 10% of the papers that are being published these days in AI besides being very complicated technically, share the code. So this means that they basically are rendered useless and they cannot be adopted by other researchers. This is a big problem. Then there's also a problem of confidence about these technologies because we lack benchmarks and we lack case examples, relevant case examples where people can learn and really trust these computational technologies. Okay. On top of that, AI is such a new field that we really lack skilled people in many of the laboratories, and this is perceived as a huge bottleneck, right? If you are a laboratory, an experimental laboratory, and you don't have a data scientist, basically, you are really not gonna start using AI methodologies because it's just overwhelming. Why? Because many of the models are perceived by the majority of people as black boxes, right? Input, output, and nobody really knows what's going inside these models. So today, I'm gonna focus a lot on this second aspect of our mission, right? Which is bringing AI methodologies or accelerating the adoption of AI methodologies around the world. AI methodologies, of course, there's a big hype about them, especially in the field of drug discovery, because they hold promise for accelerating the process significantly. Okay, and the way we could summarize how AI researchers we claim that we can help the drug discovery process is by first reducing the time that it takes to discover a drug, and second by incrementing the number of molecules that we can consider during the drug discovery process. Okay, so both axes, in principle, AI would have significant contribution. Of course, I hope I don't sound naive here. I recognize that the drug discovery process is incredibly complicated, right? But just for the sake of simplicity, and please bear with me, I'm going to simplify the drug discovery process with these 6 steps that we all know. First, a conventional drug discovery process starts by discovering a target. This might take 2 or 3 years, depending on the case. Once the target is identified, people perform in vitro screenings in order to identify some hits or some leads. Then once a lead is identified or a couple of leads are identified, this might take a year or 2, depends. Once a hit or a lead is identified, you want to optimize this lead, okay, to increase specificity and potency. This might take from 1 to several years. Once this is found, you still need to consider ADME and toxicity properties of the hits and do some more medicinal chemistry. And then the difficult part of the clinical trials and the post-approval starts, okay? So AI, basically, there has been claims in the literature that AI can help accelerate this process in each of the steps. For instance, in the target discovery, AI methodologies have been suggested to help prioritize disease-related genes or to assess the drugability of target candidates. In the lead identification, for instance, AI has been applied to optimize chemical libraries, so they are more efficient in finding hits. In the lead optimization, you might have heard about these methods. There are generative methods that starting from a hit compound, they can produce, they can invent lead molecules that might accomplish or optimize the properties of the hit compound, etc., etc., etc. Going to clinical trials, for instance, you might apply methods that might be able to predict drug-drug interactions, anticipate adverse events, etc. Okay, so there's big hopes. There's big hopes that AI methodologies can be applied to this pipeline and basically help us accelerate this process and make it hopefully faster and cheaper. Okay. So of course, as you might imagine, I have listed here a lot of AI methods. As you might imagine, each of these methods has its peculiarities, etc. But I would say that a majority of them can be simplified by this scheme. Okay, and if there is one slide I would like you to remember today is this one. Okay, is this simple scheme of machine learning. Okay, what is happening here? So here I have put compounds that have been assayed in a growth inhibition assay against E. coli. Okay, some of these compounds experimentally have been found to be active, like the first and the third, and some have been found to be inactive. Okay, so in machine learning, what we do is we take the structures of these compounds, we apply a first function that is called featurization, that basically represents these structures as numerical vectors. Okay, then once we have done this, we obtain a representation of the compounds that the computer can understand. And then we try to relate this matrix or these vectors that are related to the compounds to the actual activity. And this is done by means of a fitting function, typically a neural network, okay, which is the typical or the most popular class of algorithms that we are using these days. Okay, so there's a featurization of the compounds and there's a fitting of the compounds. Why is this so interesting? Because then you can come with a new compound like this one that has a certain structure. You can featurize this compound, obtain the numerical vector, and by applying the function that you've learned here, you can make a prediction. Okay, in this case, because we are all chemists here, you can recognize, or you could guess that this compound has a very similar structure to the two active compounds. Therefore, it's going to have a similar numerical vector to the two active compounds, and therefore it's going to be predicted to be active. Okay, this is what machine learning or supervised machine learning, supervised because we learn from experiments, machine learning is all about. Now you might say, well, this is very similar, or this is what we used to call it structure-activity relationship modeling. Yes, indeed. Indeed, machine learning in drug discovery most of the times is applied to the traditional tasks of structure-activity relationship modeling, or some people call it ligand-based virtual screening, because basically you learn from the known active compounds, yeah, ligands typically of targets. Okay, so this is the general framework of AI. So I hope here I'm demystifying a little bit the field already, right? It's just another wording for structure-activity relationship modeling many of the times. What are the difficulties, or what are the problems of this kind of a scheme? The first one is this function here. Yeah, this function here, the power and also the drawback of AI is that this function here can be very, very complicated. Okay, and the fact that it's very complicated means it has a lot of parameters. And if you have a lot of parameters, you might overfit. When you overfit, this means that you can memorize your input data, but you lose the ability to predict for new molecules. All right. So in practice, what happens with AI methodologies, because this is so complex, in practice, what happens is that you tend to need a lot of data to train the algorithm, so that you don't overfit. Okay, a lot of data because you want to commensurate with the number of parameters that you have here. Okay, this is the first real problem of AI methodologies, and this is why AI is becoming so difficult to generalize to new problems. The second challenge in AI is interpretability, right? Which was something that in structure-activity relationship modeling is key, right? So, well, a medicinal chemist might say, okay, I got a prediction, but I want to know what are the parts of the structure that are relevant for my predicted activity, right? Because I want to do medicinal chemistry on top of it. So going back from here, from the prediction back to the structure, because all of this process is so intricate, can be difficult. Okay, so interpretability in AI is really a big issue, and is something that we need to put research on. Okay, because otherwise, it's handicapped. The usefulness in drug discovery is handicapped because we really need interpretability so that we can do medicinal chemistry on top of it. The third problem is applicability. We have trained on a family of molecules, on a series of molecules that they are all very related. They belong to a certain region of the chemical space. Okay, some of them were active, some of them were inactive. So we learned a model that hopefully works very well in this region of the chemical space. But when we come with a completely new molecule, a molecule that is outside the applicability domain, we are basically not going to be able to make a good prediction. Okay, so delimiting this region, delimiting the applicability domain of machine learning models is key to gain confidence on what we are doing. Okay, so overfitting, interpretability, and applicability, I would say, are the main challenges of AI these days, especially in drug discovery. Okay, so what is our goal at Ersilia? What do we want to achieve given this very simple scheme? Well, I've told you already that we start with data. Many of the times, data is raw data and it needs to be processed. Once we have processed this data and we have prepared it for running our algorithms, we need to train a model, as I have shown, right? We need to learn the parameters so that we have a neural network that is optimized for the activity that we want to predict. Once we have done that, we need to test and validate this model. And the way this is done, it's both internally with the data that we currently have and hopefully externally with external validations, ideally with experiments. Okay. In the end of the day, we obtain a scheme like the one that I've shown that is able to make predictions for new molecules. Now, this is not the end of the story, because once you have obtained a neural network that is trained, you need to be able to use it. And of course, we don't want to be the only ones that are using it. We want researchers around the globe to use it. So there's a final step, which is very challenging, that is called deployment, where we basically bundle all of this complicated algorithm together into an application that people can use in their browsers. Okay, and this deployment step, we believe, is key for adoption of AI methodologies. Okay, these days, data scientists and researchers end up here. They don't deploy the models, and therefore they are basically not useful to the broader community. When you have a deployed model, life becomes very easy for an experimentalist, because all you have to put is an input, and then all what you get is an output. Okay, for instance, your input is a drug, and you might get as an output the predicted targets, okay, by this network. So our goal is... Yes?
There's a question in the chat. Can I read it to you? Does the machine also predict the other physical properties of the molecules, or just the activity? And how accurate is it based on the literature data?
Yes, that's an important question. So basically, usually you optimize your network to predict a small set of properties, okay, because you don't have infinite data. Now, nothing stops you from building more than one network, right? So usually the way you approach that is you optimize a network for predicting the activity against a certain type of targets, a certain family of targets, or a certain strain of pathogens, etc. And then you might have a network that is related to the prediction of physicochemical properties. Typical one would be minus log P, okay? But you can have many others. So yes, the answer is yes, this can be done, but usually you do it separately, okay? You train one network for each of your questions of interest, okay? I'm going to go to that in a minute, in the next couple of slides, okay? So our goal is to basically provide, to complete this cycle for the community and release our models in this form, in a form that people can use. Let me show you an example. For instance, sometimes I think that John Burland gave a great talk last spring, and he basically went over the AI literature for the last couple of years. And one of the papers I think that he identified was this paper called A Deep Learning Approach to Antibiotic Discovery. Long story short, this was a great paper published in Cell last January, and where basically they were learning a neural network based on activity against an E. coli growth inhibition assay. All right. So the authors here, they learned a neural network that worked very well. They validated it experimentally, but it's not really deployed. All right. So what we do at Ersilia is we go to the paper, we curate the paper, We check that everything, that we can run the code of the authors, and then we bundle it together so that in the eyes of the researcher, they can go to our tool, they look for this paper, and all that they have to do is input a drug of interest and get an output. All right. This, for a non-expert, is very different than going to the literature and trying to figure out how to apply this model to their molecule of interest. Halicin, in this case, was one of the drug candidates that they identified as a broad spectrum antibiotic. Okay, so in Ersilia, we try to go from something that looks very intricate and difficult, something like that, and perhaps overwhelming to the researcher, to something that is very simple to use. Okay, this is an easy case for us because the model has already been trained. We just need to bundle it, okay, into this kind of application. Sometimes life is more difficult for us and we need to build the models ourselves. For instance, I was just looking for an example that might be relevant to you, but you might know, I'm sure you know this paper, Science paper 2018, large screening for antimalarials in Plasmodium berghei, I think. 500,000 compounds were screened experimentally, and the results were published. Okay, so of course it would be very interesting to build a machine learning model on top of this data, right? That is able to learn the data that was produced in this paper. So in Ersilia, we take care of this. We start from the raw data, we do all the learning and training process, we bundle everything into something that is easy so that you can input your molecule of interest, a tobacco analog, for instance, and you will get a prediction of activity. All right. So this is really our goal, to go from something that might be overwhelming in terms of data science to something that is really easy to use for researchers around the world, the globe. We are just getting started, as I said, but our goals are clear. We want to build models, like 100 models at least by Christmas 2020. Models that will regard to, as I showed already, antibiotic activity, but also human cell line sensitivity, toxicity, drug-drug interactions, target predictions, etc., etc., etc. All right. So we are building this. We call it a model hub. Yeah, it's like a database of models where basically researchers. We haven't released it yet, but basically researchers will be able to run their predictions for the models that might be interesting to them, blood-brain barrier penetration, etc., etc., etc. All right. So just let me zoom out a bit. Okay. So this is our internal blueprint, our internal scheme on what type of models we want to incorporate in the midterm in Ersilia. Today, I'm focusing a lot on compounds because I'm talking to a drug discovery audience, all right? And I hope you will recognize these edges as things that are interesting. Each edge here will be a type of models, right? For instance, we want to have models that are able to predict the interaction between compounds and genes, the occurrence of drug side effects, compounds causes a disease, yeah? The interaction or the synergy between compounds, the sensitivity of cell lines to compounds, etc. Okay, so this is our internal scheme. This is our blueprint. We are trying to build models that will be able to answer all of these arrows in this scheme of, let's say, biology or systems biology or systems pharmacology. Okay, our long-term goal is to have a platform that is basically going to be able to answer these questions with machine learning models for you. Okay. So, of course, this sounds overwhelming, especially for a small team like ours. And in the next couple of slides, I'm going to try to convince you that we are in a good position of doing that. In particular, because we start from a technology that we developed at IRB Barcelona for the last couple of years. It's called the chemical checker. Okay, and all of our modeling technology is based on the chemical checker, which we recently published it a couple of months ago in Nature Biotechnology. So the chemical checker, and going back to my favorite scheme, is related to this first step of the modeling process, okay, the featurization, yeah? Basically, how do we represent compounds numerically so that the computer can understand them? Yep. So all the work I'm going to present now is basically related to this first step of the modeling process, which in my opinion is the most important one and the most fun to work with, okay, as a pharmacologist. What happens in the chemical checker? So classically, the first step, the featurization of a compound, works like this. You can take this compound. This compound is, again, it's an antimalarial. It's called alofantrin, okay? And you can explore each structure. And when you explore each structure, you discover different features. For instance, a fluoride here, a chloride here, a double bond here, hydroxyl here, etc. Okay, these features that you discover in the structure, with the computer, you can enumerate them and represent them in a vector. Okay, and this is the fingerprint or the signature or the features of the compound. Okay, so this is what classically it's been done in structure activity relationship modeling. This is classically what it has been done in chemoinformatics or even in machine learning, okay, or artificial intelligence for drug discovery. You just try to encode the structure of a compound into something that the computer can understand. Okay. At some point, and given the avalanche of data that started to appear about bioactivity, let's say from 2005 to 2008, okay, with PubChem bioassays, Campbell bioassays, the links repository, etc., we realized that well, it's a shame that these methods they only consider the structure of the compound, because in reality we now know a lot more about the compounds, right? Because we have a lot of data about compounds, and a lot of data is publicly available about compounds. So we asked ourselves, well, couldn't we do exactly the same thing that we were doing when we were encoding the structure of the compounds and try to capture all what we know about these molecules into a numerical vector that is richer and the computer will be able to understand? For instance, in this case, alofantrin, we know, apart from the fact that it is a phenanthrene chemically, we know that it's a candidate antimalarial. We know what it does. We know that it can have some side effects like cardiotoxicity, etc., etc., etc. All right, so we know a lot from the databases. We know a lot about this compound, and we thought, well, it would be very nice to try and encode all of this information together with the chemical information into a vector that we can then use in our machine learning tasks. Okay, and this arrow here is exactly what the chemical checker protocol does. Just let me break it down a little bit for you. We are aiming, with the chemical checker, we are aiming at collecting all of this information about compounds. We start by the chemistry. We look at the chemistry. We try to capture the 2D structure of the molecules, the 3D structure of the molecules. We enumerate the scaffolds. We try and characterize the physicochemistry, etc. So there's one layer where we try to capture the chemistry of the compounds. If we go one layer more in complexity, we try to capture what we know about the targets of the compounds, that is the mechanism of action, the metabolic genes, the co-crystals that we might find in the PDB, etc., high throughput screening assays, etc. Right? So we try to collect all of the information that we have about targets of the compounds. Then we go one step further. We try to collect all of the information that we have about basically the signaling pathways and the biological processes that are affected by those targets. We can even go one step further, and we try and collect from the databases cell-based assays that we might find for those compounds. Okay, for instance, gene expression changes, sensitivity against panels of cancer cell lines, morphology changes, etc. Chemical genetics experiments, etc. Okay, so we also try to collect all of this information in our database. And finally, when we have it, and when this compound has reached to the clinics, we try and collect clinical information, which is typically very unstructured. Clinical information might be basically the indications of the compounds, results in the clinical trials, side effects, toxicologies, drug-drug interactions, etc. Okay, so all
Miquel?
of this structure, yes?
Sorry, Kelly's got 2 questions.
Yes.
Is that okay? Kelly, can I ask you to please ask your questions to Miquel?
Thank you. Sorry to interrupt. Yeah, so the first part of the question is, what are the ideal numbers of compounds to make the AI models robust? And then the second question is around SAR, whether you really consider this. So in medicinal chemistry, generally we have 2 types of SAR. There's one that you would call flat, where there is no SAR, because whatever you do, you don't really have variation in activity. So for example, whatever compound you make, they are all potent. Yeah, so that's not the SAR, because SAR must be evolvable. And then the second one, of course, is desirable SAR, where you see, that's evolving. You have a range of activities depending on the molecules that you make. And of course, even lack of activity can contribute to that. So my second question is really, to what extent do you take this into consideration, if at all, before you build AI models?
Yes.
I hope that's clear, Miquel.
Yes, very clear. Thank you. So the first question both questions are quite related, I believe. So the first question about the ideal number of molecules that you might need for building a competent machine learning model. Okay. I'm sorry, I will have to say that this really depends. It depends on many factors, but I will try to give you a number. Okay. I would say that if you are building a simple model, let's say a classification model, where you just want to say whether a compound is likely to be active or not. Okay. In this case, usually your input data set is very imbalanced, meaning that you have a few active compounds and a lot of inactive compounds. Okay. Or sometimes you just have the few active compounds and you don't know any negative data. Okay. This happens a lot when you get information from scientific papers. So I would say that in this case, if you want to build a machine learning model that is going to generalize well, and it's going to work not only in the particular series that you have built the model on, you would need at least 50 active compounds. Okay, this would be like a good approximation. From over 100, you are usually well off, so it's perfect. Less than 50, it really depends on the case and the chemical variability of your training set, but it might get difficult. Yeah, so usually when we do larger scale training, this means training a lot of models massively. We ask at least to have 50 active compounds for training. That would be my first answer. Second, about the SAR, yeah, this is very important. The fact that sometimes you have a flat SAR and sometimes really small changes might have a huge impact on the activity. In those cases, when you want to go like really fine-grained, you usually don't build a classification model. What you want to do is a regression model, right? You want to predict you want to predict the constant of inhibition or whatever it is you are measuring. In this case, I would say that if you have a set of I would say 200 compounds, that would be ideal. Yeah, with the good measures, well validated, that you really trust, that would be ideal. And again, it really depends. If there is a lot of chemical variability in your training set, then it will be very difficult for the network to discover these fine changes that might have these activity cliffs, if you want. Okay. Whereas if you have a chemical space that is very focused and well controlled, then you might be able to discover these these small changes in activity that might have huge impact on the outcome. Okay, or you might be able to discover that no changes are possible that will optimize the activity. Okay, so yeah, the answer is a bit relative here, but I hope I was clear.
Yes, thank you very much. Thank you.
Very good. Okay, cool. So I will proceed. Yeah. So we have this scheme that we call the chemical checker, where we are basically constantly every 6 months updating the database, and trying to collect all of this information for as many compounds as possible. We are now at about 1 million compounds in our database, so it's relatively big. Yep. So as you might imagine, this procedure is a huge IT task. Yes, because gathering all of this information from multiple resources and processing all of this data into something that is homogeneous, clean, and that we can trust, it's really difficult. But I'm gonna cut this very long story short, and just for the sake of simplicity, just retain this picture, right? For each molecule in our database, we have a set of signatures that sort of capture the chemistry, the targets, the networks, and the cell-based assays, etc., that we might have found in the public domain. Okay. So, for instance, this would be a very, very, very tiny chunk of our database. Here you can find, I think it's about 100 compounds. Each line here is a compound. Yeah, and you can see here chemistry signatures. Here you can see target signatures, cell-based signatures, clinical signatures, etc. Okay, you can see, for instance, that compounds that are related to psychiatry, they have a particular set of targets as expected, and they have a particular set of effects in cell lines. Okay, infectious diseases also, etc. Right. So typically, the classical solution was to only capture the chemistry. So basically, people were only capturing this kind of information with the computer. They were only encoding this kind of information, the chemical information. Now with the chemical checker, we are expanding our information about molecules all the way up to the clinics when possible. Okay, so we believe that this is in principle a very powerful resource for doing chemoinformatics and machine learning. Let me show you an example. You might be familiar with this kind of maps. This is just a projection. Yeah, each point here is a compound. I'm showing here about 10,000 compounds. If two compounds are close here, this means that they have similar signatures. Yeah, in our resource. Yeah, if the compounds regions that are red, this means that those are regions related to compounds that are chemically very similar. If regions are blue, those are regions of compounds that they have a similar chemical checker signature, but they are not chemically similar. So for instance, this region here that is very populated has compounds that they all inhibit the estrogen receptor 1. Okay, and because this region is red, this means that these compounds, they have the same bioactivity because they are all SR1 inhibitors, but they are in turn also very similar chemically. Okay, whereas this region here, or this region here, is related to kinase inhibitors, that they all inhibit the same kinases, but the compounds that are here, they are very diverse chemically. All right, so with the classical approach, you wouldn't have discovered this cluster, because these compounds are very different chemically, so you wouldn't have been able to guess that they might have the same bioactivity. But now with the chemical checker signatures, you are able to bring them together, okay? And hopefully to discover them as a cluster of compounds that are related in terms of their bioactivity, okay? There are a lot of clusters here, so there's a lot to be exploited and discovered in this map. We can even do things that are a bit more complex in terms of searching similarity searches. For instance, I've taken here three compounds that are inducers of DNA damage. These compounds clearly, to the eyes of a chemist, they have different chemical structure, yeah, but they all do the same thing to the cells. So when we look at the transcriptomic response of these compounds in a phenotypic assay in cells, they are all together. Yeah, when we take the signatures of transcriptional signatures of these compounds, they are all clustered together. They kill the same type of cell lines, but as I said, and as you can see, they have very different chemical structure, right? So the chemical checker is a very powerful tool for discovering this kind of relationships between compounds. Actually, I found here a molecule that wasn't characterized as, I think it wasn't characterized as a DNA damage inducer, but was within this cluster. And indeed, when we checked it further in the literature, we found that it might have an effect as a DNA damage inducer. Okay, so this tool, even with simple similarity searches, can lead to interesting discoveries. Something that might be more close to your interest. You know this chemical moiety, yeah. So here, I think I'm showing 3 or 4 derivatives of the cephalosporins. They all have the same chemical structure. Indeed, they all have the same mechanism of action. But when we look at their effects in cell lines, it's very different, right? Which might explain the huge variability in terms of side effects in host cells in humans of this family of compounds. Okay, and the same happens with sulfonamides. We have a group of sulfonamides here, basically the same chemical structure, or the same moiety at least, same mechanism of action, but very different effects in human cell lines. Okay. More things that we can do just by looking at the signatures, the chemical checker signatures, and performing similarity searches. One thing that we think has been quite successful, at least experimentally, is the fact that we can take signatures that are related to transcription of the compounds. Yeah, so this signature represents the transcriptional effect of compounds in cells. And we can try and compare these signatures to signatures of the transcriptional effect of antibody drugs, for instance, okay, in the same cell lines. Yeah. In this case, we were interested in finding an interleukin-2 receptor inhibitor that would mimic the effect of the daclizumab, which is an antibody drug. Okay, so basically, how we proceeded, we just got the transcriptional signature in cells of basically exposing cells to the daclizumab, and we got the transcriptional signatures of our compounds in our databases, and we just looked for compounds that might have a similar transcriptional signature to the one that is exerted by the daclizumab, and then we made the hypothesis that these compounds might be mimetics of this biological drug that is very expensive and has very bad pharmacokinetic properties. Okay. Indeed, we did that, and we believed our predictions, so we went on and did some experiments. We found in our databases 19 compounds that had the transcriptional signature that was very similar to the transcriptional signature of the daclizumab. And when we tested them in some in vitro experiments, indeed, a vast majority of these compounds were promising, right? Were promising, at least in a primary and in a secondary screening, and you can see it here, right? The dose response of IL-2 dependent proliferation of the daclizumab and also of some of our candidates. It's very clear. Not only this, but we discovered that some of these compounds were not necessarily targeting the IL-2 receptor inhibitor directly, as the daclizumab is, but then we discovered that some of these compounds might act in other points of the IL-2 dependent proliferation pathway. Okay, in particular, some of them were basically affecting phosphorylation of STAT5. Okay, which is a kinase downstream this pathway. All right, so I hope this sounds good. This sounds good and convincing. We have a database that collects a lot of data that is available for compounds, and we did the big effort of trying to encode all of this information into something that the computer can use and understand. What is the problem? Well, the problem is that the chemical space is huge, right? And the majority of the there's an unthinkable number of possible molecules, as we all know, and there's also a huge and overwhelming number of molecules that we can buy, right? And of course, for the majority of the molecules, we actually don't have data, right? So I think that only around 1% of the molecules that we can buy as medicinal chemists, for only about 1% of the molecules, we have some sort of experimental bioactivity data, right? Which is very little. Not only this, but of this 1% of the molecules, actually only a few of them have made it to omics experiments that are more expensive, right? Only maybe around 10,000 or 20,000 max, okay? And of those, only 5,000 compounds maximum are approved, or experimental drugs for which we have relevant clinical information that we can incorporate in their database. And to be honest, many of these compounds have been inspired by natural products, right? So in practice, what happens, if I have to be honest, and I have to offer a more honest view of the chemical checker, is that we have something like that, right? Where we have a lot of chemical information, we have as many molecules as we want, but when we try to collect information at the bioactivity levels, then we lack information for most of the molecules, right? And this, we realized, is a big problem in terms of applicability of our models. Take for instance a project where you want to predict the bioactivity of a new natural product, right? Where you don't have any sort of bioactivity information, then the chemical checker is basically useless, because the only thing that you will have is the chemical structure. You will not have any of those valuable bioactivity information. Okay, so in order to sort of fill or infer the missing gaps in this database, we developed something that it's this time. I have to warn you, this is a bit hardcore machine learning. Okay, we developed something that is called the Siamese neural network, that basically, I'm just trying to simplify here, tries to infer bioactivity signatures of the molecules, even if you don't have experiments for them, based on the information that you have. Okay, let's say that you have, for instance, for this given compound, you know the chemistry and the targets. You might, by using this compound and the information that you might have about other compounds, we are trying to infer what is the possible cell-based assay signature of the compound. Okay, this type of architecture is really difficult, and I'm not gonna. It's really difficult to train and requires a lot of data. So this is something that we trained over a week in a supercomputing center in Barcelona, and it was really a big modeling effort. This is a picture of the supercomputing center. It's inside a church, actually, okay, because it's kept a bit colder. Barcelona is usually very hot, so they decided to put this supercomputer inside the church. So we basically used this supercomputer for over a week in order to basically fill in all the missing gaps that we were having in the chemical checker, thanks to this very complicated network architecture that is called Siamese neural network, okay, which is something that has become very popular in the field in the last couple of years. So again, long story short, thanks to this network that we trained in this supercomputer, we are able to fill in all the missing gaps that we were having in our database with inferred or predicted signatures. Okay, of course, we try not to be naive here, and we try to measure the accuracy of these signatures as much as we want. So for instance, predicting binding signatures is usually very easy. This is the accuracy. Usually we predict binding signatures with high accuracy, but predicting cell-based assays outcomes, it's usually very difficult, okay? So the signatures that are related to cell-based assays, depending on the case, are usually of lesser quality than the signatures that are related to binding activity, for instance, okay? Not only this, but we try to identify what are the regions of highest confidence and less confidence in this predicted space. So for instance, if this would be the map of all the compounds that we have in the chemical checker, we identify here a region where we don't really believe what our predictions are giving, or in the transcriptional space, we identify a region that is really very uncertain, whereas we identify a region that is of very high quality. Okay, so we really do our best not to be naive and not to believe everything that the computer throws to us. We try to evaluate the accuracy and the confidence of every single prediction that we do. Okay, at the end of the day, we have now a full database, okay, of bioactivity signatures for every compound that we can get. Okay, so now the platform becomes applicable not only to drug molecules. Okay, you can see here that it works pretty well in identifying regions that are related to infectious diseases, identifying regions that are related to psychiatry, cardiology, endocrinology, pulmonary diseases, oncology, etc. But it can also be applied to molecules that might be less well characterized. For instance, metabolites from different species. You can see here that metabolites belonging to different regions of the tree of life belong to different regions of the chemical space, perhaps as expected. You can see some commonalities between metabolites of Homo sapiens and mice, etc. We can also focus, for instance, on human metabolites, and we can see that metabolites in different tissues or fluids actually belong to different, if you want, regions of our bioactivity space defined by the chemical checker. Or you can focus, for instance, in food ingredients, natural products, etc. Okay, so now we have a platform that we believe is very powerful to perform biologically relevant navigations of the chemical space. And now let's go back, and this is my last technical slide. Let's go back again to my favorite diagram. Thanks to the chemical checker, as I said, we have done a huge effort on basically improving this step of the machine learning process, right? Thanks to the chemical checker, we are not only representing the chemistry of the compounds, but we are representing something that is which is all the bioactivity that we have collected over the years as a scientific community for those compounds in a numerical vector that then the fitting process can learn from and benefit from. Okay, and now the question is, okay, but do you obtain better models when you do that? General answer will be, in general, yes. For instance, here you can see, and I'm just showing an example here, but of course we've tried it in many different benchmarks. I'm just showing here an example of a toxicity panel called Tox21. This is a popular toxicity panel. I think it was trained, it was developed for almost 10,000 compounds. 10,000 compounds against this panel of pathways that are related to toxicity. Okay, I think it was 12 pathways here, and we have from the literature, we have the activity of these 10,000 compounds against these 12 pathways, active and inactive. Okay, so we are just trying to basically build models that are going to be able to tell us whether a new compound is going to be active or inactive against each of these toxicity-related pathways. Yeah, and the way I'm measuring the performance here is through this method, which is called the ROC curve, that basically just tries to find a balance between specificity and sensitivity. The higher, the better. Okay, so a ROC curve of 1 means perfect predictions always. A ROC curve of 0.5, an area under ROC curve of 0.5 means random prediction. Okay, so here I'm showing the result of training a good machine learning model, a state-of-art machine learning model using only chemistry. Okay, And I'm showing also the result of training a state-of-the-art machine learning model using our bioactivity signatures from the chemical checker. And as you can see, is that systematically for most of the pathways, if not all of the pathways that we are trying to predict, we get significantly better predictive power just by changing this step, just by using the chemical checker signatures instead of a conventional chemical representation of the compounds. So you can see it here. Our prediction is systematically better, and this happens with other benchmarks that we have been trying. Okay, and you can see we have a publication in bioRxiv showing all of the benchmarks that we have done. Okay, we recently posted it a couple of weeks ago. So I think that's all. I wanted to talk to you about. Some take-home message. Remember, we are building Ersilia. Our overarching goal at Ersilia is to build the first repository of AI models that are pre-trained and are related to drug discovery, and everyone can use with no technical skills. Okay, this is our big overarching goal. The way we hope to achieve that is by benefiting from this technology that I recently developed, that's called the chemical checker, that is based on this principle of integrating a lot of data from the public databases into something that is called bioactivity signatures, that we believe is much more informative than just using the chemical information alone. With these bioactivity signatures, I have showcased you how we can use them to visualize compound collections, how we can perform complex similarity searches that might be interesting, and I hope I have convinced you that they can boost AI algorithms to state-of-the-art performance with, in principle, minor effort. And basically that's it. We are an open source initiative, so we are really looking for collaborations. We are happy to listen to what others are interested in, and we'll be very, very happy to learn more about your current research interests and find ways of working together. Thanks for your attention again, and thanks for the invitation. I hope I was clear. I'm happy to take questions now.
Thanks so much, Miquel. That was a brilliant talk and beautiful slides. Thanks so much. So we have time for one question, I think. Felix, your hand was up first. Please go ahead.
Thank you. This is Felix from GSK in Tres Cantos, Spain. Hola, Miguel.
Hola.
Just a quick question. Three very related questions, I guess, but mainly one. One, is the chemical checker active already? It can be accessible. The second related is, what are the main advantages, in your opinion, over, for example, SciFinder, you know, when you can do a search chemical structure, but also selecting the data you want to get. And the third one is, what's the output you get? I mean, are those heat maps, or is it a real number? I mean, when you mean that you get an output in which you get the transcriptomics profile, you actually get that, or is a link to a publication, or what right. Do you get?
No, okay, good. So yes, thanks for the questions. So the chemical checker, it's now accessible, yes, and what you can get from the chemical checker is basically those signatures, and the signatures for each of the compounds in each of the, we call it bioactivity spaces, right? And a bioactivity space can be, for instance, transcriptomics. So if you go to the chemical checker and you query a compound, and we have the transcriptomic signature of this compound, what you can download is basically a numerical vector that represents the transcriptional profile of this compound. Okay, so those signatures, I have to warn you that we made them very dense, numerically dense, so they are really not interpretable directly. So this means that you will not be able to basically tell from that signature which are the exact genes that are being upregulated or downregulated for that particular compound. For this, you would have to go to the original publication. Okay, so basically what we offer as a resource in the chemical checker is basically the result of all of the heavy pre-processing of the data that we did from the original publication into something that the computer can understand. Okay, computer can understand, meaning basically the numerical representation that can be plugged in directly into a machine learning model. Okay, so now the chemical checker is basically a resource of matrices where you can download those signatures for the compounds. Okay, this is very different to querying a great database like like Campbell or SciFinder, as you were saying, where you basically look for particular bioactivities, okay, of a particular compound. In the chemical checker, what we are providing is a more abstract representation, these kind of fingerprints or signatures. So it's really the experience of querying the chemical checker is really different than querying a SciFinder or something the like. Okay.
Thank you.
Thanks, Felix, for your question. So, Miguel, we're out of time today. Okay, maybe we take one more question from Stephen, if you don't mind.
No problem.
Stephen, just a quick one.
Thanks. I was just wondering if, yes, thanks, Doc. It's really good. I was just wondering, so since given we have all this data that from ligand-based SARs to help train and vectorize and use these signatures, I was wondering if you've seen any approaches where you can train networks based on crystal binding patterns to help predict and optimize and drive lead optimization efforts based on protein ligand binding patterns and things like that. Have you seen such approaches? Have they gotten anywhere?
So that's a this is something that we have been working on it, and we are actually working on it. Okay, which is basically trying to design compounds that will accomplish a certain bioactivity profile of interest. That is a certain signature of interest, chemical checker signature of interest. Okay, this is something that we believe is very interesting, right? Because this means that you might have a complex signature of like a wish list of properties that you want to achieve. You represent them as a signature, and then you try and design compounds or optimize compounds in order to achieve these signatures. In practice, this is, technically speaking, is a multi-objective problem. And as you might know, this becomes difficult. So we are really struggling in trying to apply these generative models into something that is that will be able to optimize for multiple properties. Okay, we are really struggling with that. But I mean, there are approaches in the literature that claim that this can be done, and we believe we are really confident that this is going to be feasible in hopefully in the short or mid-term. Okay, there have been approaches, for instance, applying this to transcriptional signatures, right? Trying to find compounds that will have that will elicit a certain transcriptional signature in cancer cell lines, for instance. Okay, and results are decent. Okay. But yeah, we are really not quite there yet.
Okay. Thanks a lot.
Thanks, everyone. Thanks, Miguel, again for this wonderful and insightful talk. We'll certainly have to invite you back for some more AI chats on drug discovery. So thank you very much again.